you.
Yes, now here.
It is my great pleasure to give a presentation here. The main line of research in our team is understanding of functioning of biological networks. And coincidentally, it was in 2009 that we were for the first time approached by a pharmaceutical company in order to use our models for drug development. Since then, we had some experience with that for different problems. And I believe that it is a great collaboration from the both physiology and pharmacology points of view. Because physiology allows to get some insight into the mechanism of drug action and provide some pieces of advice. And on the other hand, the practical problems provide a great inspiration for understanding of biological systems. So, what is computational physiology? The essential definition is that physiological modeling focuses on understanding of biological systems, not single enzymes, but rather enzymes as a part of the whole system. At the same time, computational physiology modeling is distinct from other types of modeling by the fact that it focuses on the mechanism-based simulation, meaning that you use real laws of physics and chemistry in order to create your equations. In contrast to approaches, then you use, for example, neural networks in order to describe something. So when can you use physiological models? Here you can see the basic pipeline of drug development. And I indicated with green several time points or several steps when you can get useful insight from computational analysis of your system. First, it is actually the identification of target. When you have a complicated network, it is not always easy to say what you need to target in order to turn it on or turn it off, or which is often the case, you need to subtly affect the functioning of the system. Then, from the point of molecular design, computational physiology is not really useful unless you have a multi-scale molecule level model as a part of your physiological model. The real points are in vitro systematic research when you have a molecule that affects target molecule, but now you need to make a transition to the whole metabolic pathway or signaling network or cell-based trials. And of particular importance is the first stage of clinical trials because you often need to consider a lot before working with humans. And a great area of application is post-approval research because in real life, you always get a lot of situations when you do not understand what happens. So a little bit of the difference between what you can call physiological and non-physiological models. I put together a wide range of models under this umbrella term, including neural networks, agent-based models, and other types of models when you can simulate something without really modeling it, without really going into details. Essentially, the physiological models are much more difficult to develop. You need to understand the system really well, meaning that first humanity needs to understand the system really well. And then you have to put a lot of effort to make this model functional. However, the good thing is that you have a fairly realistic representation of the system, and you can provide unexpected insight and very specific low-level prediction, something that what happens if you target this molecule within the whole system. On the other hand, for a neural network or agent-based models, you can work with the systems that are not really explored yet in order to create a simulation of those systems. And sometimes they have great advantages because they are essentially models that should be trained. And there is no difference between training a model in general and training a model for a specific human. But the limitation of those models is that in a model, you cannot see what happens at the low level. So identification of targets, for example, could be difficult. I will talk about a couple of examples. So one example is thrombolytic therapy. Thrombosis is a major clinical issue. Actually, thrombosis is an incorrect functioning of the hemostasis system when you create an aggregate of platelets or a fibrin clot where it is not needed. And it plays a major role in the pathophysiology of a very wide range of conditions, disorders, and interventions. Sometimes you can remove thrombi, but for example, in ischemic stroke, it is not an easy task to get inside the brain to remove the thrombus. And it is an important way to deal with it is to lyse it. And there is a single drug approved for this, which is TPA. And you can see that it is not a very successful drug. So at some time point, as a part of a large collaboration supported by Horizon 2020 program, we addressed the problem of TPA action. If we look at the fibrinolysis system, we can see that it is a cascade of reaction that functions downstream of the blood coagulation cascade. Essentially, as a result of the blood coagulation network, you have fibrin, which is actually the clot, that is that binds plasminogen that can be stimulated by plasminogen activators, leading to transition of plasminogen into plasmin and cleavage of fibrin. And it is actually a network that is regulated in time and space. Here we can see that, for example, when you stimulate clot formation in plasma at the same time with plasminogen, you can see a complicated pattern of propagation of clotting and in space, and plasmin lysis front following thrombin. In thrombolysis, you have a different case. You have fibrinolysis activated after clot formation. So we considered just the network of fibrinolysis in detail. TPA, the drug and the actually physiological activator used in treatment of ischemic stroke, is inhibited by PI1 inhibitor. It can bind to fibrin, stimulate bound plasminogen into two forms, and the active form, also subject to inhibition, cleaves fibrinogen to produce fibrin degradation products. We considered a one-dimensional case with a fibrin clot in the presence of TPA. It is also a spatial process. So the model, relatively simple computational model that we designed for this system, is a set of partial differential equations. Each of the equations has the basic typical form that time derivative, the rate of the concentration change is determined by diffusion, reactions, and also advection that is not shown here. And our first impression was that this lysis is greatly determined by TPA propagation inside the clot, because TPA is bound by fibrin like plasmin, and essentially the efficiency of lysis is determined by this binding. If you have blood flow through the clot, this greatly changed. There is efficient lysis of the clot in the presence of flow compared with the absence of flow. And it is no more limited by TPA diffusion. In addition, the critical component is inhibition of TPA by PI1. In the beginning, we did not believe this inhibition to be important because the concentration of TPA used in treatment is much greater than the plasma concentration of PI1. Unexpectedly, PI1 had a profound effect on the efficiency of clot lysis, as you can see here. And the explanation for this was, this is the lysis clot size, and this is the width of the fibrin front as a function of time. And you can see great effects of PI1 at the concentration much below those used for treatment. And the explanation was very simple. When you consider the process in space and time, the very low concentrations of TPA at the front come in contact with PI1, and so PI1 can regulate the efficiency of TPA diffusion at very low concentrations. We analyzed the effects of this in the presence of flow, and for permeable clots, the role of PI1 is very low. And this is just some further development of that study, where you can see lysis of two-dimensional clots composed not only of fibrin, but also of platelets and leukocytes. So the conclusions for the first part is that we can get some insight in the regulation of the system. We can explain inefficiency of treatment in some cases, and we can provide prognostic markers for the treatment, which are blood flow through the clot, which can be determined by tomography, and pi1 concentration in blood. And we also can have suggestion for further drug development that drugs without binding to fibrin or not susceptible to inhibition by pi1 can lyse clots that are not lysed by TPA. I would like to thank our great team of collaborators and those who supported this project. Another story that I would like to describe briefly is the problem of platelet signaling. Platelets are cells that regulate hemostatic response. They aggregate, and in order to aggregate, they need to be activated, which is controlled by a huge network, which I will not describe in detail. So essentially, what we need to know is that there are several activators, including some produced by blood coagulation system like thrombin, some released by other platelets, and some like collagen are activators that come in contact with blood upon vascular damage. And a platelet is essentially an analog computer that integrates this signal to produce a physiological response, most of all activation, but also procoagulant response. Platelet undergo programmed cell death in order to accelerate reactions of blood coagulation. And at some time point, in order to understand how this works together, we began, first of all, from the basic point of view, to simulate this system. Here you can see an example of our early model. This model describes the events beginning from the stimulation of one of the thrombin receptors in platelets, the signaling network, which is essentially a basic calcium signaling network controlled by IP3, and the first events in this network, and this is how they are represented in the model. Using this model had to be a stochastic model because platelets are small, so differential equations are not the perfect way to simulate it. So using this model, we analyzed platelet response and were able to show that overloading of mitochondria with calcium is the controlling event that leads to platelet cell death and procoagulant activity. A year later, we investigated these predictions experimentally. Here you can see calcium signaling leading to cell death. And on the right, you can see two platelets side by side, and with the collapse of the last mitochondrial, this platelet becomes dead. Later, we were able to investigate problems of patients with Wiskott-Aldrich syndrome, which undergo treatment in our center for Children's Hematology in Moscow. Wiskott-Aldrich syndrome is an inherited disorder that combines bleeding and immune deficiency. And actually, Wiskott-Aldrich protein is a cytoskeleton protein that is involved into actin regulation. Platelets with Wiskott-Aldrich in Wiskott-Aldrich syndrome are smaller, but are fully functional. And there is no clear idea why these patients have thrombocytopenia, which is the reason for their bleeding, and in most cases, a reason of their death. If we analyze these platelets under a microscope and monitor them for some time under physiological conditions, we can see that platelets in Wiskott-Aldrich syndrome undergo some kind of programmed cell death. When we loaded these platelets with the mitochondrial inner membrane potential sensitive dye, we observed the same picture, very similar to the one we observed in healthy platelets stimulated with thrombin or collagen. Calcium increase, overloading of mitochondria with calcium, and necrotic kind of cell death. We didn't have any idea of what to do with it until we used a mathematical model with several mitochondria to show that platelets with fewer mitochondria and smaller size undergo platelet death more readily because of their wrong volume to surface ratio and less stability of the metabolic network because of the fewer numbers of mitochondria. And very interestingly, the experiments confirmed that platelet in Wiskott-Aldrich syndrome undergo programmed cell death more readily if they have fewer mitochondria. And we also confirmed using cyclosporin A that this cell death is prevented by blocking the mitochondrial permeability transition pore. Another story that we when we came in contact with the practical problems is PAR1 and PAR4 signaling. These are two thrombin receptors that are very similar, but different in their affinity and lifetime, but they have almost the same signaling pathways. We used computational models in order to show that their combination provides a perfect combination of sensitivity and within a wide range of thrombin concentration and a wide range of times. So one receptor is short responses for low thrombin concentration, while other works for long-term responses and a wide high concentration range, while together they provide a combined response. Interestingly, we observed a heterogeneity in the personalized responses, which was explained by different genotypes of the donors, which we were able to find that are in line with our analysis. So the donors with a strong PAR1 phenotype have a very hill-like response over time, while donors with predominant PAR4 phenotype have a slow, long response, which is very interesting in view of the recent development of PAR1 antagonist that failed in clinical trials, which suggests that this difference and different role of PAR1 and PAR4 peptide receptors would explain this. This is the line of the model development. By the present time, we have a more or less complete virtual platelet that can now be trained for personalized patients in order to predict their responses to therapy. So this is an example of a very long-term development of a model that now can integrate the major signaling pathways and metabolic network of a cell. And as a result, we can have predictions for good target, for example, for Wiskott-Aldrich syndrome, or explanations for drug inefficiency in the case of PAR antagonists. And I would like to thank the team that did this research and our great collaborators. So thank you for your attention.
Thanks a lot, Mikhail. You leave a little bit time for questions and answers. And we have one question in the chat from Dmitry Shulga. If you have largely different timescales, you must face the so-called stiff system of equations. How do you deal with this?
Thank you. I will put it like this. In real life, you do not actually have at the physiological level too many radically different timescales. Because it is not a good system from the physiological point of view. Okay. Of course, if we try to simulate all details at the same level, we might encounter problems like this. But usually physiological systems do not have such a great difference. For example, for fibrinolysis and for blood coagulation, there are short-lived enzymes that have a time scale of minutes, like thrombin, and long-lived enzymes like factor IX that have a time scale of tens of minutes. And as for longer or more rapid processes, their in-depth consideration is not usually necessary.
Thanks a lot. I don't see more questions in the chat. Maybe I will ask you. Yeah, actually, I understand that your modeling may lead to some personalized treatment of the patients. But when we talk about personalization of the therapy, it's always a problem of economic. Is it suitable to get enough data from each person in the real clinics, or it's maybe for just some selected persons, like minister or so?
I believe that the answer to this is a combination of advantages compared against price. For example, if you need to perform in-depth genomic or proteomic analysis, and then a lot of bioinformatics, then you need a lot of reasons to use personalized treatment. However, I didn't tell today about one of our projects, mostly because it is confidential. But at some time point, we had a contract to develop models of glucose metabolism in order to personalize treatment based on the readings of continuous glucose testing. And in this case, the price of analysis is very cheap, while advantages are quite significant. So this is something that could be used on a mass scale.